right, these notes are actually going to go into your AP Bio spiral notebook, not the comp book, but the spiral notebook, because these notes are a little bit more intense. We've got a little bit more that goes along with them. All right. So, starting with this diagram. So I have drawn a cell. It's a beautiful cell. Within the cell, I have a nucleus. Within the nucleus, I have DNA. And this is going to make more sense here in just a minute. Outside of the nucleus within the cell, I have a ribosome. This will make more sense in a minute. And here I have a polypeptide or a protein that's being formed. Transcription occurs in the nucleus. Notice I've highlighted that green. RNA processing occurs in the nucleus. Translation occurs at the ribosome of the cell. This is protein synthesis, and this overall generalized steps of protein synthesis means we're taking DNA, we're taking a gene, converting it into RNA, and then making a protein from that RNA. So DNA to RNA to protein is protein synthesis. The template strand is the DNA strand that provides the pattern or template for ordering by complementary base pairs, for ordering the sequence of nucleotides in an RNA transcript. A non-template strand and template strand, which one is the non-template strand and which one is the template strand depends on the gene itself. But the template strand is the one, the DNA strand, that is used to make RNA. Transcription, RNA processing, translation. So this is protein synthesis. The term the central dogma means protein synthesis. It's called the central dogma because protein synthesis occurs the same way in all living things. And so central dogma, or the central overarching concept of protein synthesis. So on my diagram before, I highlighted transcription green. The steps of transcription are initiation, elongation, and termination. I highlighted RNA processing orange. In RNA processing, we have 5' and 3' caps that are placed on the DNA. We have splicing that occurs, and we have ligase. So the ligase is going to glue things together. In translation, the important parts of translation are messenger RNA codons, mRNA codons, that's messenger RNA, transfer RNA anticodons, tRNA, transfer RNA codons, and ribosomes. And ribosomes are going to bond amino acids into a protein. A gene is a segment or section of DNA chromatin that codes for a protein. The promoter is the DNA sequence on the template strand where RNA polymerase binds. TATA, the TATA box, is a DNA sequence within a promoter. This DNA sequence, the TATA box, it tells RNA polymerase where to bind. The terminator is a DNA sequence that ends transcription. And then the ribosome is a cell organelle. It essentially reads the messenger RNA transcript, reads the messenger RNA message, and it bonds amino acids together to build a protein. Transcription. Script, scribe. A scribe is somebody who writes. A script might be, something that has writing on it. Transcription is the message. During transcription, the messenger RNA message is created from DNA. So DNA is the template, and then the DNA is used to write the message, transcription. The first part of transcription is initiation. The RNA polymerase binds to the promoter region. Then the DNA unwinds and the RNA synthesis begins on the template strand of DNA. Next is elongation. So the RNA strand is created and it is built 5' to 3' with RNA nucleotides. Remember, RNA nucleotides, they don't have thymine. We have uracil instead of thymine. So the RNA nucleotides are A, U, C, G. A pairs with U, C still pairs with G, or bonds. A bonds with U, C still bonds with G. Finally, in termination, the RNA transcript is complete and it's released from the DNA template strand. So this picture helps to show on the blue, that would represent the DNA. The red would represent the RNA. You would have your primer where the RNA polymerase, which is represented by this big blue guitar pick looking thing, would bond. Then, as these nucleotides are added, we elongate, we make the RNA strand, and then finally, termination, this piece is separated from the DNA. Next, we have RNA processing. So inside the nucleus, the DNA is transcribed. A message is written. But that message isn't perfect as it is. It needs editing. So it's like you writing sentences or writing a story or something. You have to go back and run it through spell check and edit it and add punctuation or change the spelling of words, etc., etc. So RNA, that message needs to be processed as well. It still happens in the nucleus. So within the nucleus, the messenger RNA is edited or processed. Okay, so you have this little strand of DNA. We've unzipped it, and one strand is going, or one half of it will be the non-template strand, and one half will be the template strand. So in this situation, for this particular gene, this part right here is the template strand that's going to be used to make the message, to make the messenger RNA. This one's a non-template strand, so it's not going to be used. All right. My messenger RNA is created. So C, G, G, C, T bonds with A, T bonds with A, T bonds with A, blah, blah, blah. But when I get down here, T bonds with A, there's no T in messenger RNA. So A, U, C, G, T, A, C. So those are bonding together in that format. So messenger RNA is created from the DNA template. Whoops, sorry. So this is DNA. This is messenger RNA that's created from the template strand. All right, there's a particular reason for the colors on each one of these. Anything that I've highlighted orange is an untranslated region, or abbreviated UTR. So this is an untranslated region. This is also an untranslated region on the other end. These three codons. Oops, gotta fix that. All right, there. Okay, these three codons are an exon. The next four represent an intron. These two codons, exon 1, 2, 3, 4, 5, 6, 7, represent an intron. 1, 2, 3. That's going to be another exon. So make sure you color and or highlight appropriately because it's going to make a difference. So first four are untranslated region, next three exon, next four intron, the next two exon, the next 1, 2, 3, 4, 5, 6, 7 is intron, the next three exon, 1, 2, 3, 4, another untranslated region. Okay, so now we have a specific molecule called a spliceosome that splices. Introns are trash. The introns are cut out and removed from that messenger RNA strand. Exons are expressed. So exons end up being glued together to be the message. Ligase is the enzyme that glues exons together. Introns are trash, they're cut out. Exons are expressed, they're glued together to make the messenger RNA strand. And just for your information, ribozymes, so spliceosomes cut the messenger RNA, but ribozymes are introns that splice themselves out of that messenger RNA strand. All right, so after things have been spliced out, oops, and hang on, I forgot I had an actual visual of my introns that I cut out of my second strand. I cut them out and I put them over here and reminded myself that introns are trash. So this one gets taped in here, and then you've got a second one that you're cutting and removing the introns and taping together, gluing together the exons. Okay, so now that we have the introns and exons where they need to be, now we need to protect our messenger RNA strand. A 5' cap is added. It is a modified guanine, GTP. A 3' poly A tail is added. Poly means many. A stands for adenine. So the 3' poly A tail, that's 50 to 250 adenine molecules that are added. These not only protect the ends of our messenger RNA, but they help that messenger RNA strand to bind to the ribosome. So on this messenger RNA strand, I have cut out the introns that are trash. I've added my GTP, my 5' cap. I've added my poly A tail. And now I have my untranslated regions, I have my exons, and I have my 5' cap, 3' tail. This messenger RNA has been processed and is now ready for translation. Translation, the messenger RNA is now changed into a protein. So here is my messenger RNA, and I made it a little bit smaller so that I could fit it all on here so I could see. Here's my 5' cap, I just colored it on there, 3' poly A tail, just colored it on there, but then I have the rest of this here. Untranslated region, so that's not important. Untranslated region, so that's not important. So what do I have right here? The exons are what's important. AUG is my first codon. In translation, we said that the messenger RNA codons and the transfer RNA anticodons are important. Transfer RNA is going to bring the appropriate molecule to match with our codons, and it will be carrying an amino acid. For us, when we translate our proteins, we are going to use a codon chart. Here's a codon chart with letters, and then here is your codon chart with actual amino acids on it, and this we'll be using in class a little bit more. But this is just kind of giving us an idea of how this is working. So my first codon is AUG. I come down here to my codon chart. A, U, G. I look to see where they all match up, and I find that AUG codes for a start codon, or it codes for the letter Y. A is my first letter, U is my second letter, G is all the way on the bottom, that's my third letter. So that's why this says start codon. Next, we have AGA. AGA, so A, G, A. I look to see where all three of those come together, and it comes together at the letter B. So that's why there's a B here. G-U-G, so G, U, G is an I, and then go through the rest of them to see if you can figure out where the rest of those letters are coming from. These would represent amino acids that the ribosome would bond together, little peptide bonds between each one of them. And then this word would represent the protein that was formed, or the polypeptide. In class, we mentioned the fact, or I mentioned the fact, that there are only 20 different types of amino acids. Those 20 different types of amino acids make thousands of different proteins, and they do that simply by rearranging the letters. The cell cannot understand DNA. So DNA is not a language that the cell understands and can follow the directions. DNA is changed into messenger RNA. The cell cannot understand that language either. The cell cannot understand messenger RNA. The genetic code needs to be translated into the language that the cell can understand. That is a protein. The cell can understand the language of proteins. So if all we have is DNA, nothing is going to get done. Even if we change DNA into messenger RNA, which is a message, the cell still can't understand the message. It's still a foreign language. Once we translate these languages into protein, the language of proteins, now the cell can understand and can follow the directions and do what it needs to do. All right, so here is a little diagram. Here is my messenger RNA. 3 nucleotides represent a codon. So the 3 letters on the messenger RNA is a codon. The 3 letters on the transfer RNA is the anticodon. These transfer RNA molecules are carrying those amino acids. Once they come here and line up, the ribosome is going to put a peptide bond between the amino acids, and then you're going to have this protein chain that's created. Once the transfer RNA adds its amino acid, then it gets kicked out and it goes back and it picks up another amino acid. Okay. The codon, set of 3 nucleotide bases on messenger RNA. tRNA represents transfer RNA. Transfer RNA molecules bring the appropriate amino acid to the ribosome. The anticodon on the transfer RNA matches with the codon on the messenger RNA. Transfer RNA does not make the protein. Transfer RNA is just kind of like a shuttle vehicle. It's taking the amino acid to the ribosome so the ribosome can make the protein. Remember that a polymer is made up of monomers. The protein polymer is made up of amino acid monomers. The polypeptide may then have additional folding and structure changes based on the amino acids it's made of. And we kind of talked about that in class, that if it was hydrophilic, hydrophobic, positive, negative, those amino acids might want to come together, or they might fold to be exposed to the outside, or hide on the inside. So it depends on their characteristics, how that protein is going to fold. And finally, protein synthesis doesn't always go the way that it should. Sometimes there are mutations. When we think of mutation, a lot of times we think of something bad, but not all mutations are bad. A mutation is only a change in the DNA. That's it. Mutation is not a bad thing. Mutation is not a good thing. A mutation is very simply a change in the DNA. The mutation is not bad unless it is harmful to the individual. So ways that it's harmful is if that mutation results in the death of the organism, or if that mutation renders the organism infertile. Those are the only ways that a mutation would be considered bad, is if it's harmful. Types of mutations. Let's just do this first. A substitution mutation is also called a point mutation. Point mutation is a change in one single nucleotide base. One base is swapped out. Point mutations can be silent. A silent mutation is a change that has no effect on the phenotype. So, maybe it was changed out with something that ended up with the same amino acid. Point mutation can be a missense mutation, changes one amino acid to another. So we had an amino acid, and now this missense mutation changed it to a different amino acid. There could be little effect or a big effect depending on what the new amino acid is. So if the new amino acid is similar, there might not be much of effect. If the amino acid is very different, it could completely change the protein. A point mutation that's a nonsense mutation it's going to change a codon into the stop codon. And that means it's going to stop making that protein early, and you have a protein that's not going to do its job, a non-functioning protein. Insertion or deletion mutations are either going to add or remove. Insertion adds, deletion removes. They're going to add or remove a nucleotide, causing a frame shift. All of the downhill codons are changed. So you take one out, everything past that is changed. You push one in where it's not supposed to be, everything past that is going to be changed. And finally, a mutagen is a physical or chemical cause of a mutation. X-rays can be mutagens, UV light, carcinogens are chemicals that can be mutagens that can cause mutations.